Axel. Oh, yeah. So yeah, wow, I'm humbled. Thanks, Axel, and thanks, Christoph, for the introduction. Yeah, so today I'm going to talk about stuff that we did in Berlin. My lab is actually in computer science, right? So most of us are computer scientists. We do have one bee biologist, and we have engineers, and I think Pranav is also here. I saw him in the guest list here, in the participant list. But most of us are computer scientists, you know, looking at data, recording data, building training models, trying to understand what's going on in a beehive. And as you said, Axel, this is a beehive is a mess to make sense of it. And yeah, I thought I could share some of the things that we did and that we are currently doing, and maybe even a little bit of what will be coming out soon. So let's get right into it. Most of what I will be talking about will revolve around the honeybee dance, which was the start of, or let's say the end of my pure computer science life and the start of my biology life back in, I think, 2008 or so. And so I will be talking about stuff that we can do with software to detect and also to map dances, like Mackie Kuvion already talked about, and also other behaviors that we may be interested in understanding. You know, the waggle dance has received so much attention, but I think there's so much more going on. So maybe we can help in sharing this. And then moving up to the colony level, we have developed a system with which you can track all the bees in there, all the bees that carry such a marker, and all bees that do not. And then we can try to make sense of that data. And I also saw Tim Gernert in the participant list here. He is a friend who has already developed a similar system. So it would be great to talk about similarities or differences between the two systems. And so I then will lead over to understanding individual roles of bees in the context of this social network, let's say. And now my slide has changed by itself, so it's good to have someone to kick my behind. So just a quick recap. I know you should be all familiar with the waggle dance. So when a bee comes back from a food source, you see that behavior. Let me just switch to my, to my pointer. Oh, no, that was wrong. Here. So we see that bee that just came back here performing this weird behavior. And then there are other bees, and I have marked only one, that follows kind of the position and also the angle in a similarly stereotypic fashion, right? And we call this one the dancer and that one a dance follower. And now the cool thing is, and that was so crazy back in I don't know, 2008 or so when I learned about this, it's not just that the dance shows properties that correlate with food source location properties and food source profitability. It's also that bees that follow the dance can decode this information. And what is encoded? Well, so you have basically a polar coordinate. So distance and direction encoded in the dance. So if you have a reference outside in the field, the sun's direction, and the angle with the food, you can see dances with the bees waggling in a direction that has the same angle with respect to gravity. And if the sun then travels further over the sky, this angle changes and the dancing bee rotates accordingly. Yeah. So, and since we humans can easily decode the dance, we can write software to do that for us. And why we would do that, I think that was part of Maggie's talk. I think she was the first in the series, right? But I did that because I wanted to build this robot here. So we wanted to understand exactly how this robot should move to be able to ask a couple of questions, like, for example, what kind of cues do we need to implement in the robot? What kind of cues do these followers need to actually start following? And so this Mark B here is one of those followers. We had, I think, 200 or so not individuals, but 200 instances of bees following. So that could be a lower number of bees that were following, but may have been following multiple times. And we also were able to track recruits in the field with a harmonic radar, but the number of recruits and the number of dance followers compared to natural dances was much lower. And the question was always, okay, why? And why is it so variable, right? So when we would start, sometimes bees would, jump on the robot like instantly and follow beautifully, like in this video. And sometimes the robot could move for days and nothing would happen. And so I never understood after my PhD what was the reason. And so there are many, many more questions why one would try to understand the waggle dance even more, and dance followers especially, why they would start following or not, right? But the first step is to not improve the robot, but more or less improve our understanding of what's going on. And the first part of this is understanding the waggle dance itself, detected automatically. And that is the first paper that I wanted to present to you a little bit. So how can software detect a waggle dance? Well, you just need a very bad camera. And this one is like a cutout window from a $10 webcam on Amazon. But this webcam, PlayStation camera, delivered 100 hertz. And so if we look at one pixel here, which kind of touches the position where this waggling bee goes through, you see that the brightness, changes accordingly, right? With the frequency of this motion. And so, our thought was, okay, let's just detect these periodic, developments of the brightness of this pixel over time. You usually can do this with something that's called Fourier transform or wavelet transform. So what you basically do is you have sine and cosine waves at certain frequencies, and you just put them over this function, and then you take, let's, for example, say the dot product or some kind of similarity measure, and then you can optimize the position, and then you get a score of how well that matches. And so that's what we basically did. I'm simplifying a little bit, but in the core, that's what we did. And then we can, move this over. And if we find many of these pixels that in a sufficient amount of time show a good match, then we have a detection. And that's what the software does. And then to determine the angle, we use difference images, and so consecutive images, in time are subtracted, and then we can take the Fourier transform of that, filter a little bit, and then we have the angle. And that was the first software solution that we published in 2016 or 17, I think, 17. And we used that to decode dances. We measured and evaluated exactly how good the software can do that. Fernando was the PhD student back in the day. He's now working in Italy. As you can see, although the software is really precise, so it has a comparable precision to human annotations, we have a fairly large spread around this feeding site or this food source here, this green triangle. And this is the location of the hive. And we mapped all the different dances, even the natural dances, but also, of course, the dances from the foragers that we trained there. And so this is a tool that we thought may come in handy for others, and some labs in the community have tried to using it. It's still a gap between, I think, like technical people and the actual users, the biologists that want to use it. So we kept improving this, and David, one of the next generation PhD students in the lab now, and this is the link here on GitHub, has uploaded a new version that can also work on higher resolution videos, so you don't have to run it in real time. You can upload your videos. I think we are limited in terms of frame rate to, I think we have to have at least 50 or 60 hertz or so. So you don't need 100 hertz, but you need 50. Still more than most of the people have, I think. I talked to people that had 25 frames per second videos and were sad that we couldn't use the software on these. But yeah, recording new dances isn't taking too much time, so we just have to wait for summer. Okay, so now the question is, okay, what can we do with the software, and how can we understand the questions that result from these data sets? And the first thing that we did is to just look where dances happen in the hive, and this is something that we already know. There is a specific location in the nest, and the nest here is just a one-frame observation hive, so it's a tiny nest. It's only 2,000 individuals, but still these can survive the winter and flourish and rear-brood. And something that we don't or didn't know yet is something that we will publish soon. If you train bees to different food sources, and we used two sources that are 180 degrees or so apart, we see that the dance locations are separated. And it is a very noisy signal, but we have enough data, right? So we don't need human annotations to detect dances. We just run the software over months of dances. And so this will be published soon. I can't really give you more. Other than this, the software can now not just detect waggle dances, it can detect shaking signals, it can detect ventilation, it can detect tremble dances here, and different vibratory behaviors that, yeah, I think deserve some attention. And I would love to hear your opinion about whether that's useful for you or not. And I will come to my vision, let's say, of how we can use machine learning and software and computer science tools in the study of communication behaviors, for example, in the honeybee. But so for now, just a little teaser, there will be another paper coming up soon. And I think the method is yet ready. It's a little slow for real-time processing. So David is trying to improve it in terms of real-time runtime. And so this is just a little, like a little story arc about the waggle dance. But the real questions are how everything is embedded in the social interactions and the social network of the hive. When we had our robot, and I would see some bees following, I would always ask myself, so why does this bee follow the robot and not the other bee? Why is it that a bee follows right away, and why does it take so long? So for those bees who would follow, I would have loved to know more about them. So are these certain patriline, or do these bees have some experience with a certain scent that was on the robot or not? Or did they already have some experiences with the robot before? And so what I really would have loved to have is a system that I'm going to show you now, which is basically giving me or allowing me to, send a query to the database, give me everything about bee number 1, 2, 3. Yeah, and it should cover probably, in the best case would be the whole entire life and everything that the bee has done and everyone the bee has met. And so I could run analyses, see what the following behavior in the future correlates with in the past. Yeah. So the first step that we did, and this is also done by was also done by Fernando, the first PhD student I showed you, develop these tags. And these look a little different than the ones Tim Gannett and his group used. These are round and curved, and they are so because we wanted them to last as long as possible. And we had those queen tags, right? And we knew they would, stay on for years as long as the queen would live. And we tried these and find similar durability, let's say, even for foragers that fly out and collect nectar and pollen, or in hive workers who inspect cells and have like a lot of mechanical stress over the thorax. No? And so this is what it looks like in the colony, shot with a normal HD camera. But this is what we do to record our data. So we used to have 2 cameras. Michael Smith, who I think should also be here, at least he told me, he gave us 2 new cameras, and now we have 1 camera per side. And this is what it looks like. The images are synchronized with infrared flashes, so they can't see it. And you see that those tags are nicely visible. The resolution is 12 megapixels per recording, and we record 6 frames per second. So that's as much as we can encode videos for. So we do that on the fly and push every video that we make. I think it's covering a couple of minutes or so of hive life, and then we push it to a network storage where it's safely mirrored and backed. So and this is what we do for sometimes 10 weeks or so. Michael and Constance did that even over the entire winter. And so it's just a matter of resources. So if you have these resources, you can run this forever. And this also includes the resources of human labor to actually get those tags on, but this is another story. So the interesting part now, software-wise, comes next. So these raw recordings are then fed into a pipeline of different stages where we first detect the location of each of these tags, or even untagged bees or bees that are inspecting cells. We then look for, or we then identify those bees. So we have a machine learning model that gives us the ID, but also the spatial orientation, 3 angles of rotation. And so this is what the raw data looks like. And then we track these over time. Another machine learning model that connects consecutive locations because those IDs could be slightly wrong, and it's not as easy as just connecting the same ID. And then we have short tracklets, and then another machine learning model connects these tracklets to long trajectories that, in the best case, are as long as the life of a bee. And then everything is uploaded to a database, and many scripts can then look at various properties of this behavior, for example, whether there's food exchange between bees, or waggle dances, or dance following, or other things. Yeah. And so we have published everything open source. You can go to this link, which basically lists everything that we did. which includes also an app for the biologists who sit on the feeder and wonder what and who this bee is. And so you take a photo with your mobile phone, and then the app tells you who that is. And it's not just numbers. Benjamin Wild, another PhD student, has come up with another machine learning model that generates bee names that may sound like Kirby or Barbie, but it has to be a bee pun. Otherwise, it won't be a valid bee name. So I haven't told you about the stuff that we can do with this data. I think I should tell you that there's too much that we can do with the data. And there's only one thing that would fit here in this talk, which is the question about, okay, what is the bee actually doing? And there are different levels of detail or like scale, temporal scales, let's say. And on one of these scales, this question relates to the task a bee has, for example, being a nurse, so caring for the brood, feeding the brood, or processing nectar, or collecting nectar and pollen outside in the field. So these are task categories. And what we know about bees, so the textbook tells us that, is that the age roughly determines this task. Yeah? So young bees become nurses, then as middle-aged bees, they become nectar processing bees. And then as they age over, like, let's say 21 days or so, they become foragers. And we already know that this isn't a one-way street, and young bees don't always necessarily have to be only nurses. So there's a lot of variation, but this is pretty much the rule of thumb. Yeah? So one number tells us exactly what a bee is doing. Now, the cool thing about bees is that they are pretty organized spatially, let's say, right? So we have a part of the nest that is only used for the brood, and a part of the nest that the bees use for honey storage, and another part of the nest, as we saw before, for communication dances. Yeah? And so if you look at now time and space, it's when bees emerge from the brood, they are in the center of the nest. And as they age and the next generation emerges, they are pushed out, becoming middle-aged bees in this intermediate area. And as they age further, they become, nectar collectors, so foragers. They come in and unload food to the middle-aged bees. So it kind of roughly makes sense, also spatially and in time. And now, since bees do that, and we know that they do that, we can use that to just look at the positions to classify a bee as a nurse or a nectar processing bee or a forager by looking at those profiles, right? So we can just look at all the positions that we have detected with our system and look into human labeled regions, whether that is, whether the bee was in the honey storage area or in the brood area or the dance floor. Yeah? And so this is great in bees. And our question in the following was, so what if we had a system that would not have such a rigid, spatial organization, or would have an unknown spatial organization? What could we do to determine the task of an individual, given the only thing that we have, which is social interactions? So positions in an unknown system wouldn't give us more, but we would know who's doing what with whom. And that is exactly what we did in this study. So we used the tracking data to come up with social interaction matrices, and they roughly looked like this. So for an interval of time, let's say a minute or an hour or a day even, we could look at all the interactions, and these could be variably defined. So it could be food exchanges or could be dancing. In this case, it's really dense. These are proximity matrices. So whenever a bee and another bee are sufficiently close, we would count, the duration of this interaction or the number of times they do this in a certain interval of time. And then since we have a lot of data, we could do this for many non-overlapping intervals. And so if we look at one bee here, the life of that bee is basically described by this vector or this row in this matrix. So all the interactions with the other bees over time. And so these, given the number of interactions that we had, I think we had defined 11 different interactions or interaction types, would create a data set for one bee that is 11 million numbers per bee in day. And that is a lot. And that doesn't help us in understanding what's going on. And that's why we used a compression algorithm that we developed for these matrices and came up with a one-dimensional number per bee and day. And since we formerly used age to determine task, we now call this new number network age. And if you're interested in how we did this, I think we just used two well-known algorithms. It's not even deep learning or anything AI or what. It's written in the paper. We can talk about this later. Just for now, it's a compression algorithm, and it gives you one number. And if you look at the spatial distribution of bees with that number being low, we see that, they're all in the brood nest, which is something that we would expect also for, biologically young bees. And that's also the case in biologically young bees. But if we are getting older, there's much more spread, much more variation. But in network age, so the middle-aged or the middle network age bees are in this middle region here, and the old ones are on the dance floor, which is a much clearer and much lower variance picture of what we saw in biological age. So using network age, we can predict with more than twice as, or we can predict the task or the presence profiles of individuals more than twice as good as biological age would allow. And since it's hard to actually know the task of a certain bee for all different tasks that there are, we trained bees to a feeding site, which makes them foragers. So this is easy for them. And we looked at the distribution of network age and their biological age. And in the biological age, we see, I can see the axis. I have to move my window. I can't read it otherwise. We see that even very young bees, down to the age of 5 days or so, up to very old bees, fly out and apparently have become foragers. But in network age, this is all around the age of 30 or 40, the network age. So this means we were able to extract information from the social network that did not rely on absolute location information to learn what these bees are doing. And we can also use multidimensional network ages. They get better in the prediction of also other properties. So for example, the number of days until they die, or whether they die in the next week or so. So this isn't possible using the biological age, but especially we believe due to the fact that network age predicts also foragers much better, and foragers are the ones that risk their lives outside. This prediction gets better for death prediction, but also, although not strikingly well, we can predict the time of the biggest activity peak for that individual. We can also predict other parameters that I'm jumping over in the interest of time, but we can talk about that later. Curious thing is that network age over the biological age seems to split early on in life, so that we have a bimodal distribution of, looking at the biological age bracket of, now this window, I can't even read it. I don't know, 20 or 10 to, I don't know, 20 to 30, yeah? We see that there's a part of the distribution of bees that have high network age. So these are the foragers, and other bees at, in the same age, biological age bracket, have not yet become foragers. Yeah? And that is interesting because it is not that it's same age cohorts, right? So these could be the older ones in a way, or these could be the younger ones. We've looked at same age cohorts. So for example, in this example, 123 bees that emerged the same day. And then we track them and their network age, and we see that this blue cluster here starts developing into high network age early on in life. And then a second cluster becomes foragers later, and the rest of them still only slowly increases the network age, so changes their task. Yeah? And it's unclear why that is. This paper was focused on the method, and we haven't yet found the time and money to actually look at this. But I found this really intriguing because it's unknown what determines this shift. Is it genetic, like are these Petri lines, or is it some type of social interactions? We don't know. So I think this is very, very exciting. And this is the paper that we published last year, I think. And these three people here, bright students, Ben, David, and Adrian, were the first authors on this. This is a joint work with Ian Cousin and Michael Smith when he was in Constance, and Kirsten Trainer. And Dirk Brockmann, by the way, he works in COVID modeling, but he has a knack, or some interest in social networks, and we borrowed Adrian from his lab. Okay, there's a follow-up work I wanted to share with you. This was published on a small conference. I think we undersold it a little bit, but Benjamin wanted to start working outside the bee world, has been with me since 2010. I think he deserves as much credit as I for this lab we are working in. And he looked at another method, how to create such, let's say embeddings, right? So numbers or vectors that are lower in dimensionality than those 11 million numbers that we had. And looking at the interaction matrices over time, we see that since new bees are emerging and old bees are dying, these matrices can look very different. And any method that tries to factorize them has a problem, because the meaning, the semantics of these embeddings, of these factors that we are extracting, isn't guaranteed to stay the same. Yeah. So a vector for a bee, or a network age number for a bee at a certain point in time, here and earlier or later, if that's the same number, it doesn't necessarily mean that they have the same task. And that is something that is the biggest point of criticism for network age. And he came up with a solution that looks like this. So we still want to approximate the interaction matrix shown here for an individual J and I. This interaction strength now needs to be factorized into two vectors, one reflecting the individual I in her, specific age. So this is an old bee and has a certain vector that looks like this. And this vector times this vector then should approximate the interaction strength. Yeah. And individual J now could be a younger bee and has different vectors, or different values for these three components of this factor vector. Yeah. Now, the underlying model that we train is based on two components. One is a mean embedding, a mean, like mean curves for these three values that are learned, and so-called basis functions, so deviations from this mean development. And so the model tries to minimize the amount of, or is encouraged to use as few basis functions as possible. And in the end, one of the best models that we trained actually manages to describe the interaction dynamics over time with only two factors. One is the blue, F0, and one is this orange factor, F1. And it kind of also already shows us what we already know, right? So in the beginning of their life, they start, and then gradually, and there is something going on at the age of 10, right? So when in network age, so the first cohort went up to become old and foragers, something's changing rapidly in both of these factors, and then there's only gradual movement. And these are 4 different randomly sampled individuals to just show you how much variation is in there, how even powerful the model is with using these offset basis functions to have some differences from the mean factors. Yeah, we can look at these individual lives through the eyes of these two factors, and they make sense. We measured it similarly to how we did that in the network age paper. So, for example, high values of the orange factor mean the probability of presence in the brood, or the proportion of time in the brood, is very high. Yeah? So that means these are young bees. Yeah? And if we have low factor strengths in this factor, which is here, these are old bees, they are never in the brood nest. Yeah? So this is just another way to visualize what we already saw. And this makes sense. We can predict the task. We can do whatever I showed you with network age. Also with this, it's called TNMF, temporal non-negative matrix factorization. It's a nerdy term, but you can read about this here in this preprint. And the good thing here is that we can now compare bees that have never overlapped in their lives, and we can compare bees that have never even lived in the same colony. And this is the remaining gap that we had in the network age paper. And we could also use this model to look at intraday variation, so circadian activity. Yeah? So this is a model that Ben trained, I think, with a resolution of half an hour or maybe 1 hour or so. And you see nicely that there's a lot of variation early in life, but even more strongly later in life. Yeah, maybe I should just look at these things here in the interest of time. You can cluster the individual factors over the age, yeah, into different, let's say, developmental groups. Yeah, so these or almost 2,000 individuals start much differently from the mean factor 1 or factor 0, F0, And this one looks very different, but these are, let's say, clusters in life. And we now have to look at exactly what that means in terms of behavior, because this only reproduces or approximates the social network, right? Okay. Yeah, so I think the vision I am trying to convey here is that it would be great. I mean, from my point of view, I'm a computer scientist, this is so obvious, but I have learned that biologists often don't think that way. So I want to track everyone, detect all the, or almost all the behaviors that we know of over the entire life of an animal, and then see, you know, who's doing what, with whom, where, how long, when does it start, when does it stop? Do bees exist that never dance, or that never follow a dance, right? So all these things have been described in textbooks, but never quantitatively, right? So this is what we need to also exactly understand what is natural variability. So what, yeah. And I think the way a colony reacts to different environmental conditions to, you know, bring it back to like a stable cycle is something that we have no idea about how this actually works. So my vision is that we can take these data sets and train machine learning models to represent this data, right? And then look at these models and look at the data to understand what are relevant factors, create hypotheses for the biologists to test. It doesn't make sense to record everything and then try to solve everything in this data set. I think this is the first step, you know, to create hypotheses that are based on the data. But there are many challenges, and so although we have, you know, substantially improved software, because we heard that these things are hard to actually get going, right? So to install it on a computer, you still need technical skills, or us to help you, and that is something that a small lab like mine can't do for most of the things that I would like to do. And it's really tedious work still. I mean, it's not that automation gives you more time. It actually takes more time to set everything up, because you still have to do manual work, marking the bees, training the bees, and all these things. So so I would estimate that, and this is not bad. Oh, it should be a D here, magnitude. We need an order of magnitude more money and people. And I think this isn't that bad. I mean, it's not a moonshot project where we need hundreds of millions or billions of euros or dollars. I think this is something that we could, as a community, achieve in a couple of years if we had the money. Okay, so that was what I wanted to share with you. Thank you for your time and for your interest. This is based on the work of Fernando, of David, of Ben, of Adrian, 4 of the many students in my lab who have created an amazing amount of data, of papers, of software. I'm indebted for life for the work that they did. I love these guys. Thanks to these especially, and also thank you, Christoph and Axel, for the invitation. And yeah, now we can talk questions.
Yeah, thank you, Tim. That was a great talk, and kind of a little bit, a lot of stuff to digest. Let's kind of see. Okay, are there questions?
We have a couple in the chat, but let's maybe start with in-person questions, if there are some.
Yeah, so I can also, there's one hand raised. Yeah, Olaf.
Yeah, good morning. And thank you for moving the time of this presentation 2 hours later from Western Canada. It's 8 o'clock in the morning here. So I'm really appreciating the time and the talk that you gave, Tim. I have several questions. One is just directly related to your sort of estimation of $2 million or 2 million euros at the end there. And as you know, we've contacted you and we tried to set something up here on the cheap when I was still in the US, and it didn't work and falter. So one question is, you know, what are those costs then that you foresee to set it up in like for a real project?
Yeah. This is a rough estimate, right? So I was thinking, okay, what does it need to get this vision done? And we already recorded, I think 4 data sets, maybe 5 or so, over 8-10 weeks or so, 6 to 10 weeks. And that is cool. And we improved the recording and everything looks fine, I think, for the last 2 data sets. The first ones were learning how to improve things. But it took us, like, we started in 2013, and that was solely funded on scholarships of these students. So there was not one project grant involved in this. And I think now we are ready to actually record something and to get this data. But then we only have one setup. So we have to buy a couple of setups that we run in parallel. And we have to run a couple of experiments to manipulate, to see how they react to certain changes in conditions. And Tim and Gene Robinson have done quite some work in this, and maybe you can share something of some of your experiences, Tim. I think you know, if you have many parallel setups, you have to mark all those bees, so it takes more time to actually tag every bee, and it takes more manpower. But even if we had that, it takes a lot of effort to actually get your hands on the data, because it's not easy data. It's a lot of data. It takes a lot of time to actually just load it from the database. It's simple. It sounds simple, but like a query, give me the positions of one bee over her entire life takes sometimes a day.
Right. So it's not super expensive equipment necessarily. It's mostly manpower. And skill labor that you are counting in this. Okay. Yeah. If I may just ask one biological question real quick, I know that there are other questions, but you showed these clusters of bees that transition towards foraging from in-hive activities. And I was curious whether you tested, I mean, I always thought that the age of first foraging or that transition is a continuous variable, and yet you showed distinct clusters. Is that statistically supported, that there are different groups? Or how did you test this clustering?
No, we haven't tested this. This is just like a byproduct of the network age paper. To be honest, I can't tell you exactly how many cohorts we had, but I would guess, like from my memory, it was 20 or so, 20 cohorts, 20 days in which we introduced new bees, always in the range of 50 to 100, 120 or so bees per day. And we always saw clustered transitions. Now, whether these are significantly different from something, I don't know, a baseline condition, we haven't tested that, I think.
Thank you.
Okay, so who wants? Tim? You raised your hand?
We can't hear you, Tim.
Hi. Great talk. Thanks. It's always fun to catch up with your work, and really enjoyed listening to it. I had one question regarding the tracking of bees. You, in passing, mentioned a couple of times, or it sounded like you're also able to track bees that are unmarked, they don't have a barcode. Now, as someone who is tag thousands of bees. I know how hard that work is, and nobody likes to do it. So that's why my ears perked up. Are you able to track bees that have no marker? That'd be really great.
So yes, we are. I can't tell you how well that works. So David did that. I know. Is David here? I invited him, but he has a little son, so maybe he didn't. No, I can't see him. So, so the way we do this is we have a machine learning model that receives features of, let's say, a candidate detection. Like a focal detection and candidate detections in the next time step. And this model, predicts probability of association. And for unmarked bees, one of the main features, the ID, is missing, but it's not the only feature that we use. So I guess the quality of this mapping of this association is lower than for the marked bees, but I can't really give you numbers here.
Mm-hmm. Okay. And if I can ask another question, I was really intrigued to see how when you showed the network age graph for the blue, brown, and green groups, how that one group, the brown one, started to increase its network age after a couple of days. And I was wondering whether you looked at the size of the blue group, whether that shrank, so that maybe due to mortality or whatever reason, there was sort of a vacancy in a certain task group that got filled by the brown group then.
You're saying this one, yeah?
Yeah,
so
that transition.
I remember that as like 2 years ago, maybe 3. I found that so intriguing. I pushed the PhD students, David and Ben, to get something out of this, but in the end, nothing was conclusive. So it looks like the blue ones are less, right? So that some of them may have died and then pulled them up. And that was one hypothesis that we had, but I think nothing came out of this analysis conclusive. But I think this is something that we can now address, right? So, and I just wanted to share that there's so many curious things, even for the waggle dance that is known for so many decades. There's still so many new things in there. And yeah, I would think about new things like network age. There's even more unknown things to see. Yeah.
Right. Thanks for the talk again, and good luck figuring all of that stuff out.
Thank you. Let's catch up later in Berlin sometime.
Okay.
And then there's a question by Anthony James Nealman.
Hello, Tim. Great talk. Thank you so much for sharing your work. You briefly mentioned on a slide that one of the other researchers had developed an app that identifies bees by tag simply by taking a photo with the phone.
Yeah.
Is that technology that's readily available, or can you talk a little bit more about that?
Yeah, it's so the app was developed by a student called Aki, and the model that gives a name to the number was developed by Ben. So I wasn't precise here. And this can be downloaded under this GitHub link. I think it's covered now. You can't see it, but I will share these maybe under the YouTube video that we then upload.
The same that's at the end of the tracking all members of a honeybee colony over their lifetime paper? I think I saw a GitHub link at the end of that one.
Let me just post that in the chat here.
Yeah, that'd be great. Thank you.
Oops.
Yeah, very, very cool to see. And some of I think there was a paper out of France recently that RFID tagged bees and came to a similar conclusion, at least about that some bees never leave the colony and shift age very early. So very cool work. And the tagging, the printing of the tags, or the making of the tags, is that relatively straightforward and also something that's readily available?
Yeah, that's what Michael can tell you, because he perfected our method. So Michael, you're raising your hand, so maybe you can start by answering that question, and you can ask your question yourself.
Wonderful, thank you so much.
Yeah, thank you. Michael, are you here?
Yeah, I'm here. Can you hear me?
Yeah.
Let's see. Yeah, so I guess, I mean, first, great talk, Tim. It's always nice to see what you're up to. In terms of the tags, I would say it's pretty readily available. We use some never tear paper, which is like a plasticky, like composite paper. And then there's like a small metal piece that you need to have the curvature on, and then a hand punch. You can also use it with a drill punch. But I think if I got down to it, I think I could do like 100 tags in about 15 minutes. So it definitely gets to the point where you can do it pretty well. You do have to find an old school printer that won't smear the ink. But this is all, as Tim said, relative to the other issues, that is a very minor one to get those things going. And it is cool because you can actually print kind of any tag combination that you want. So I definitely, especially with the phone app, it's actually, if you're needing someone to be just like, you kind of want to shoestring this, it's very doable. I think at the larger scale though, I think Tim's right. It does definitely takes a lot of human power to do these things. So actually my question, Tim, was actually related to, so when people talk to me about like, oh, I want to get an automated tracking system going, I kind of compare it to a microscope. Like imagine the first microscopes that were invented, and they provide all these amazing opportunities to observe these things, but these microscopes require a lot of specialized skill and labor to build and create a microscope. And I wonder, in your lab or members of your lab, or, I could imagine something kind of like a lot of universities have core facilities where you can, send your samples and, all you get is the genotype back, or, oh, I need to get this sequenced or anything like that. Could you imagine a world where we would actually have things like, like an automated bee centralized location where people could say something like, okay, I have these 2 patch lines and I want to track them for the first 20 days. Because there is so much work that goes into acquiring these data, but it's not necessarily something that every single person has the resources or the needs or the desire. And so you could actually say something like, okay, Olaf, you want to test some compound against how it changes their behavior. And you could actually contract it out to someone, Tim, that would have, like Ben would have been the perfect person to start like, the bee microscope project where he could, basically take orders for people and then say, okay, I can give you a specialized data set that really would not exist otherwise. I think the caveat on that though is that then you'd also have to do the data analysis side. So it's like, it really can end up being a mess.
This is the most time-consuming part, right? I think tracking everything that could be done as a service. I don't know. Do you want to start a company?
Oh God, no.
Yeah, I was always thinking, nah, I don't want to do that. But in a way, I don't know. I think at some point maybe there's something valuable that we can like sell as a product, but I was never thinking about that, and I can't even imagine how big that market is, right? So how many groups would pay to get this
and also,
data?
I mean, for your lab, like, I mean, you have lots of questions you want to answer, and you don't necessarily, like the microscope analogy, you don't just want to make microscopes all the time. You want to do stuff with microscopes. But yeah, it's very cool. It's always been great to work with you. So if you do decide to have a student, if David and Ben decide to start up their own company, just know that I'd probably contract them out for a couple things.
Yeah, it's bad that they're not here. I think Ben is traveling and David has a son. Yeah, that's too bad. But yeah.
Well, great talk.
Okay.
So nice to see you.
Thanks, man. Thanks.
Sorry, Michael. Okay, Vidor.
So I was wondering if this can do the individual tracking just as well if you mark bees with colored dots. Like, I guess if I gathered right, what you are doing is you are filming them constantly in infrared so that you don't have a visible light signal that's disturbing them. But if one is just doing some dance experiment and for that duration using a visible light signal, then could this do just as well with like following sets, like triplets of colored dots, or does it need this kind of symbolic tags?
I think there's a paper by Anna Dornhaus's group, and they did that with ants, if I remember correctly, with colored dots, so sequences of, I think, 3 or so dots. And that's an algorithm that was published, I think 2014, if I remember right. So obviously it's possible. The decision that we took for those tags was because we want them to fly out and come back, and then we would be able to re-identify them. But yeah, if you know there's only, I don't know, a group 1 and a group 2, and you have a certain coloring system, then yeah, absolutely, that would work.
Okay, Christoph, do you want to read the question from the chat?
Yes, so there are a couple of comments and a couple of questions. A first comment by Tapovan Farms, just saying this is very interesting. I'm interested in using the software and participate in any beta program that you may have. And then there's a link with an email address that you can look up in the chat if you want to get in touch.
Just copy that.
Then we have a question that is, out of the 123 bees, if you had removed the early foragers in blue, would that minimize the delay in the maturation of the late foragers in yellow?
We don't know, we haven't removed them. So that's exactly what we have to do, right? So new experiments focusing on these hypotheses.
Mm-hmm. Then another question, did the difference between early and late maturation foragers correlate with environmental change?
I think we recorded definitely weather parameters. And I'm pretty sure we looked at correlations. But yeah, so since this wasn't the focal question, we never came up with a conclusive theory of what's going on. Maybe environmental parameters. Sorry, can't give you more.
That's all right. Another question or another comment first. Fantastic work. Thank you very much, Tim, for sharing these. I am wondering if your method could differentiate pollen foragers and nectar foragers, since pollen and nectar are kept at different parts of the hive. I would imagine that this might be possible.
So there's like two software components. One is detecting dances. I think the new version could be improved to be able to differentiate between nectar and pollen dances. And then there's this tracking system, which runs at a much lower frame rate, right? So we could, in theory, test that if we had labels on the bees, right? So for a certain interval in time, this bee is a nectar or a pollen forager, then we could try to see whether features like network age or other features like space use give that away, but we haven't done that.
Okay. Another question is, with such a detailed interaction tracking and extensive marking of individual bees, have you found any evidence that bees have friends, in the sense that do they have small social circles with whom they would like to go out to work and hang out and dress? Mm-hmm. I guess that's a bit related to an early slide that you have where you show that bees visiting different feeders are in different places.
So that was one of the hypotheses that I had when I asked myself, okay, what's going on? Why do only certain followers follow the robot? Do they have kind of preference? And do they either, recognize the smell of the food, or do they have a preference for the location on the comb, or a preference for the advertised portion of the environment where they had maybe been before and had acquired memory that makes it easier for them to locate the food or whatever? So there's a lot of things that I was thinking could be possible. And we did a couple of experiments to decide whether it's like a social preference or like a location preference. And these were inconclusive because the experiment we did, and we have to, and we will do this summer again, just gave us too little data. So the training of the bees was hard. We were carrying bees from one feeder over to another feeder to drink from this feeder and then fly back and dance to the new one. And then we would see whether in group bees that were used to fly this way then would follow the dances to this feeder, but carrying them over on a bike, that was just too much work. And now we are trying to use tunnels for that, which come with other particularities. But what we do see is that if you have a group foraging at one feeder, they will be close to each other on the comb, in the dance floor. And they can be separated spatially, and that is for sure. The question is, well, there are many questions. So how is that working? So what is the mechanism? Do they find each other, or do they find certain locations on the comb surface? And we come up with a theory in this new paper, which is a simple mechanism, let's say. And I think we won't be able to answer the question whether the followers actually anticipate this. So, and use this information. But I think, this will be follow-up work. And it's pretty cool, cool paper that David is preparing now. Will be published, I think, in a couple of weeks as a preprint. So,
so I vaguely remember, and you probably know this better, that von Frisch talks about these groups in his book, that they're resting together. So I 15 years ago since I read this, so I don't remember if he made any suggestions about how the grouping exactly comes about.
Yeah. I looked it up in the book when we prepared the paper, and he said nothing about dances. So it was just that they hang out with each other. I think that's a different aspect, and we haven't looked at that. But when it comes to dancing and dance locations, yes, we can see that, that division.
Okay, should we maybe switch to in-person questions for a bit? I think.
Yeah, go ahead, Abby.
Hi, can you hear me?
Yep.
Yeah, great talk. I really liked the work and the paper. So I had a couple of questions. One is a little bit, I mean, it's slightly related to the work, but a little more abstract. And that was about using these proximity networks, to recapture how complex a task can be. I mean, the obvious tasks for which proximity networks work well, like say prophylaxis, where you have like very obvious interactions, but maybe for some other tasks, these networks are not so good. And I was wondering if you've given any thought to that, and what so you mentioned that you had used 11 different tasks, and I was wondering if they were all equally well represented by proximity networks, or if there are some that are good enough to be used, whether some are sort of okay, but still this is the best we can do.
No, those 11 networks were pretty different. Not all of them were uncorrelated with the proximity network, but much sparser. For example, the prophylaxis network, for example, is much sparser, and we have other networks that we call speed transfer networks. So if two bees interact spatially, like come in relative proximity, their motion speed after this interaction may change, right? So relative to their own speed before the interaction. And that difference in own speed before to own speed afterwards, this difference is the network link strength. So it's not symmetric, right? So one link describes how I change when I meet you, and the other link is how you change when I meet you. And what else do we have? Yeah, so yeah, we had the different interaction definitions, but I didn't get your question on that. Sorry.
Yeah, I was trying to, my main question was, because I also started doing some tracking and stuff, and I was wondering how well proximity networks can, recapture the breadth of behaviors that we see in, say, a hive, right? Because obviously there are some instances where it works well, but there are some behaviors for which it doesn't. And if you sort of use your whole tracking based on these proximity networks, then you're getting a somewhat biased view of what the colony is actually doing at any point in time. And that then affects your results and your inferences. So I was wondering if you've given any thought to that, and whether you have any solutions in mind that you're working on, or if you think proximity networks are the best we can do right now.
No. Obviously, there's much more informative stuff that we could detect, like dances and dance following and stop signals, which are very fast. So we wouldn't even be able with this system to see these shaking signals. The queen alone with her signals. So there's so many things that we could potentially detect and then formulate in social networks. But again, this is a lot of work. So maybe we should share everything and then build up upon one system and try to, I don't know, find people in the community that have tech skills, and then, add component per component. This can't be done by one lab.
So I have another question, if I may. Yeah.
Mm-hmm.
I was thinking about the network age, and and then later in the talk you touched upon this. You were talking about how it's specific to the network itself, right? And it might be, so my initial thought was it might be difficult to use this as a metric to compare across colonies. And then you discussed this new work, which I was not aware of till now, or you were using some non-negative matrix factorization to sort of look at a value that can then be used across colonies. I'm wondering if you have data from multiple colonies where you have the same factors, say factor 0 and factor 1 that you talked about, and if they show similar patterns, because that would be essential to sort of state that, now we can go across colonies rather than just be colony specific values, which may not be easily translatable to.
Well, I mean, we do have something, but the question is how advanced that analysis is, and nothing that we would be able to publish, I think. Partly also because Ben is away, and but we do have data recorded in Constance with Michael, which is or was a much different colony. So it had 3 stacked comb frames on top of each other, right? So a 3-frame observation hive. I don't know how many bees were in there. Michael, maybe you can add that, but so it's much different colonies, and I think Ben trained a model on that one too, but I can't tell you whether that is advanced enough to actually make something out of this. Yeah.
Okay, thank you.
Okay, Christoph is.
So there was just one comment by Michael. He said up to 6,000 bees in the Constance observation hive. Then there was one other question. First, thank you very much for your wonderful talk. Is it possible for your method to re-identify the same waggling bee with the green rectangle on the video in the first several slides? I guess that refers to the one that you showed at the beginning.
No, that was my master thesis back in the day, and that wasn't able to identify bees. We do have 2 classifiers today. So we have this low resolution but high frequency video feed classifier that isn't able to identify bees, just that is a dance, or that is a shaking signal, or that is whatever. And we do have a classifier that uses the trajectories. So this can detect dances also very well, but the direction component, the decoding, isn't as as good as in the first feed. So we have to integrate different data sources.
Okay.
Okay, Christoph, then I have a question. Yeah.
Go ahead.
Tim, so I mean, I'm not sure whether I understood everything, but so if you kind of like in your data found out that there is a shift at day 10, right? Network day 10 or whatever it is, then it reminds me on changes in JH hormone titers that kind of like make it kind of are involved in kind of like the change in behaviors. So my question, do you think that your system can predict what the bee will do? So the other way around. So if you find kind of like these changes in your models, but then you can do the models only at the end of the bee, can you kind of like come up with prediction that you can say that at this age 10 or 12, this bee, you know the trajectory, and then one can catch that bee and, for example, do hormonal measurements or kind of like other measurements to see kind of like what could be the physiology underlying kind of like this changes in the life trajectory.
I think we did that, and we were fairly good in predicting the future network age, which is the future task. Yeah, but the sampling, right? So this is what's missing. We didn't do that, so we didn't interact with the colony except after I think 3 weeks or so. We replaced the substrate because we didn't want fresh unmarked bees to emerge into the colony. So every 3 weeks we have like an interruption of a few hours where we had to replace the comb. But yeah, that's exactly one of those things one could do, right? So what are the factors that determine this behavior? So this could be memory, learning and memory, could be hormones like juvenile hormone or whatever, or could be even, I don't know, epigenetic stuff or microbiome, or you name it. There's so many things under what we can access here, and my lab has no microbiology or gene experience, so we would need someone to help us there. I think Gene Robinson and Tim Gannett, they're onto some of these things, and I think you published also something. Tim, I remember something. Maybe you can add.
Okay, so there's also something interesting. So when you kind of like in the beginning of your talk, when you were talking about the spatial kind of distribution, right? First in the brood area, then something in the middle, and then something in the dance floor. And that reminded me strongly at kind of one of the early kind of gene expression studies in Gene's lab, where they found that there's a difference between day olds, 4 to kind of 4 days old, 8, and then foragers or something like that. So there was, I mean, that were the only groups that they tested. But there you could also see in the gene expression pattern that there was a change, right? Mm-hmm. So that kind of like, I mean, so let's put it this way, kind of your figures reminded me on that paper. Yeah. So, and where you have then this transition.
Mm-hmm. I mean, yeah, these are candidates I would put my money on as well, or some of my money.
Yeah. Gustav, is there anything else?
Yeah, Christoph, you're muted.
There is currently nothing in the chat. I don't know, Tim, if you have time for another last question.
Sure, yeah.
I think Vidur would be the next. I guess Ebi's hand is from the previous question.
Okay, so so first I just need to quickly get clear what I can infer from the answer to the previous question. So So I guess can I infer from your answer to my previous question that that I can take the module from Anna Dornhaus's group's code that tracks ants based on color dots, and can I take that module of code and insert it into this, and then have that identification by colored dots module, and then will this software like track the dances of those bees just as it would the bees tagged with this monochrome thing here?
So I don't know if the paper that I mentioned by Anna Dornhaus's group, whether they actually share the code that you have to check. But if you would be able to get your hands on this, sure, you can track the dances with this software. So you don't need any stuff that we did here to get the motion data for the dances. I'm not so sure if I understood what you're interested in doing, but yeah. So our software for identification needs those tags.
Okay, I see. Okay. Okay, it is clearer now. So what my work at present is in the distance signaling part of the waggle dance. Yeah, but I am given to understand that that there are other softwares which can follow the motion, but for the specific purpose of like tracking every behavior of the bee over its entire life, this code needs the specific kind of tags you have designed.
Exactly. I don't know if so a good question for you to ask yourself maybe is, what parameters do you want to extract, and do you actually need to track a bee? Maybe you don't need to track a bee if you're just interested in the duration of a waggle run. You can use other things, right? So the one software that I presented that runs on high frequency video, that doesn't give you an ID, but you can do that afterwards by hand, for example, or I don't know. But yeah, I'd be happy to discuss this if you like. Yeah.
Okay, thank you.
Sure.
So I didn't, if there's a bit of time, just one more little, I forgot to ask it in this conversation. So yeah, so I do need IDs. And now the next question is, what happens? I guess this doesn't happen in mellifera much. You're like able to keep them in an observation height such that like basically everything everybody does stays visible. They don't like hide under other bees, etc. But maybe I've ended up using like colonies with too high a density, but Serrana might also do that. So with this Apis Serrana, they tend to be hiding under other bees and a lot. So many waggle dance runs are like hidden. And then so I was wondering, so of course, like when the start or the end is completely hidden by like other bees of a waggle run, then of course that has to be discounted. But if a waggle run starts and then is hidden from view by some other bee, but it ends again visibly, can this software like record that as a waggle run?
I mean, I can't give you a guarantee that it will find, or I can't even give you the guarantee of the opposite. So it has to be tested. I don't know what it looks like. And so in the end, if a human is able to detect a waggle motion, so nowadays these deep networks that we train can match human performance. So if you are able to, like, right, a rule of thumb is, if you are able to recognize and detect dance that is partially occluded or so, the software will be able to do that as well.
Right. fair enough.
May need to be retrained and fine-tuned to do that with new data, but still
okay.
possible. No?
Thank you.
Okay.
Thank you very much. I have no questions in the chat anymore. So I would say we thank Tim again for a very interesting talk, and all the participants for a very interesting discussion.
Thank you for having me. That was great.
Yeah, great talk.
And then I hope to see many again next week. And to everybody who is left still, have a nice evening or rest of the day, or a good night for Axel.
Yeah,
okay.
Thanks.